Hello. Today I'm going to be telling you a little bit about the underlying scientific principles of DNA sequencing. It could well help you to review some basic aspects of techniques such as the polymerase chain reaction, or PCR, before viewing this recording in more depth. And there is a PCR animation on the 3095 gateway page. Ultimately, DNA sequencing is very similar to the PCR reaction, and it's based on the fundamentals of complementary base pairing. So here we have a short DNA sequence, C, T, A, G, and whenever there's a C on one side of the molecule, it will be joined by a G. Wherever there's a T on one side of the molecule, it will be joined by an A, and so on and so forth. So guanine, adenine, thymine and cytosine, those are the four main bases. C's join with G's, T's join with A's, and vice versa. So DNA sequencing, as I say, is very, very similar to PCR. Standard DNA bases, also known as deoxynucleotides, have a binding point, a hydroxyl group attached to carbon number 3. That's the OH, an oxygen and a hydrogen atom. With the sequencing reaction, we use a slightly different form of DNA base. Rather than a deoxynucleotide, it's known as a dideoxynucleotide. The fundamental difference with a dideoxynucleotide, show it a bit further, there is no binding point attached to the carbon 3. We just have a hydrogen, not an oxygen and hydrogen, so no binding point on carbon 3. And we call these dideoxynucleotides. In the sequencing reaction, the dideoxynucleotides are actually fluorescently labeled. The C is fluorescent blue, the T is fluorescent red, the A is fluorescent green, and the G is fluorescent yellow. Hopefully you can just about see that G there. That's fluorescent yellow. So G is yellow, A is green, T is red, and C is blue. The fundamental difference about the dideoxynucleotides: after a dideoxynucleotide has bound, there can be no subsequent elongation of the DNA molecule. So ultimately, they will inhibit subsequent elongation, subsequent piecing together of DNA. So, for example, in the sequencing reaction, it's just like PCR, and in fact we have a single primer, whereas PCR has two primers, so that's a slight difference there. With the sequencing reaction, we have a single primer rather than a pair of primers, okay? And we have a mixture of both fluorescently labeled dideoxynucleotides and normally labeled deoxynucleotides, so normal A's, T's, C's, and G's. The other fundamental principle of DNA sequencing, we have to imagine in the sequencing reaction, we're working with a PCR product, so there will be well over 1 million copies of our gene. Let's say we want to read this sequence. We want to decipher the sequence C, T, A, G. Okay, C, T, A, G. And that's what we sent away to the sequencing company to be deciphered. We pop in our fluorescent dideoxynucleotides and our non-labelled deoxynucleotides, and we heat up the reaction just like PCR. We then bring it down to the primer annealing temperature, and let's say in this particular permutation a C comes down and joins with this G. Because it's a dideoxynucleotide, a blue C, all subsequent binding on the molecule is inhibited. This means we're left with a fragment of one base pair in length. But elsewhere in the reaction, let's imagine that in this instance we get a normal binding, a normal C binding, followed by a dideoxy T. Okay, in this instance the reaction will be terminated after 2 base pairs. And all other permutations have the potential of occurring, and indeed will occur. So for example, in this example we have a C-T normal bases binding before the inhibitory A base takes hold. So in this instance we have a molecule of 3 bases. Then finally, in this final example, we have a C, a T, an A, then finally we have the fluorescent G joining here. And so in this example, we now have a molecule of 4 base pairs in length. So that's what happens in the sequencing reaction. How then do we decipher the genetic code? How then do we decipher the sequence of bases in our gene of interest? We use a technique called capillary electrophoresis. Capillary electrophoresis, very similar to standard electrophoresis, whereby shorter molecules of DNA travel further through the gel than larger equivalents. So at this point I should inform you that it could well pay to re-familiarize yourself with the principles of simple electrophoresis. But this is capillary electrophoresis. And with capillary electrophoresis, we use a glass capillary, a glass tube with a very fine bore running through it, and within that bore, the central hollow tube running through the glass, we have cast into it an electrophoresis gel, a standard agarose gel. And we put our sequencing reaction into the top of the capillary, and we run down using standard electrophoresis. Okay, so we run an electric current through the capillary. Okay, as I said before, the shorter sequences of DNA travel further than the larger sequences. So our 4 base pair sequence ending in G, our 4 base pair sequence, will travel the shortest distance down the gel, then followed by our 3 base pair sequence, terminating in A. That will travel to this point in the gel. You can see where the green line is there. We then have our 2 base pair fragment terminating in T. That will travel even further down the gel, and we can see the red band there. Finally, we have our 1 base pair fragment ending in C, and because that's only 1 base pair in length, that will travel all the way down to the bottom of the capillary. Then the capillary is bombarded with laser energy, causing the dideoxynucleotides, the labeled bases, to fluoresce. So here we see a point of fluorescent blue, red, green and yellow. And by an understanding of what colour our fluorescent bases are, C's are blue, T's are red, A's are green, G's are yellow. By looking at the pattern of fluorescence in our gel, we can decipher the sequence of bases in our gene. In this instance, G, A, T, C. And that's the fundamental principles of DNA sequencing. Thanks for your attention.